we'll be discussing some questions on computational metagenomics. So first question is, the Permanova test statistic is based on the ratio of between group variance to within group variance. Then which of the following is not a repeat type in microbial genomes that one can face while doing metagenomic sequence assembly? So not a repeat type in microbial genomes is DNA A region. Which of the following is not a step of genome assembly pipeline? That is functional annotation. So we have cleaning up data through pre-processing. We also have building sequence graphs and traversing all graphs to recover genomes, but functional annotation is never there. So that's the answer. Now, fourth question, select the one which is not a metagenomic assembler program. That is Picristy, because MegaHit, Metaswades, and MetaVelvet all are metagenomic assembler programs, which I've discussed in my previous lecture. Picristy is not, so that's the answer. Now, the CheckM tool that computes the completeness and the contamination of newly assembled genomes uses a certain kind of genes for its predictions. These are basically SCGs that are single copy marker genes. Now, refer to the output of strain plan for a few species as noted below. This is the strain plan output given here. Which species shows the maximum within host variability? So the answer you can see, this one shows maximum variability. As you can see, the box plot is also varying, and we have outliers a lot, reaching the non-polymorphic site rate as 80% and crossing that as well, right? Crossing it to this point. So that is this fecal bacterium prosnitzi, right? Which species has the least within host variability? So you can check from that. This is the least one. That is this one. Okay, pretty easy question. Which of the following option is correct based on the two statements below? Strain plan is a read-based approach that profiles the single nucleotide polymorphisms (SNPs) in the clade-specific marker genes of each species or clade. That is true. Statement 2, it can be used to perform function-level investigations of the strain variations. No, that is false. So that is the answer. The figure shows the variability in different strains of a species across different geographic locations. Which is the species that shows most prominent interstrain variability across all geographical regions? You can see a lot of variability seen in PC1 of fecal bacterium prosnitzi again. So that's the answer. Which of the following shows the most distinctly partitioned strain-specific clusters? That is this one. This one. Eubacterium rectale. In meta-MLST, new alleles are assigned if they differ by no more than n number of SNVs compared to any of the reference alleles. The value of n is 3. Now, which of the following is a point in denoting the origin of replication? That is Z. This is the origin of replication. And the peak to trough ratio of read coverage will indicate how fast a species corresponding to that genome is responding. Moving on. Moving on to our next question. A random forest is trained to predict whether a patient has disease X based on certain biomarkers. Table below shows the feature importance scores for the top 5 biomarkers used in the model. If we decide to only keep biomarkers contributing to 80% of the total feature importance, which should be retained? So you can see biomarker A, biomarker B, and C. When we add up, we get 0.8. So that is our answer. Now, the topological overlap measure (TOM) is widely used in WGCNA, that is weighted gene co-expression network analysis, to assess the similarity between two nodes or genes based on their shared neighbors. Consider the following statements. So TOM incorporates both direct and indirect connections between nodes. That is true. A higher TOM value indicates stronger topological similarity. True. TOM is only calculated for directly connected nodes in the network. False. TOM values range between 0 and 1. True. So the false statement is statement 3. Moving on. Write very short answers or fill in the blanks. So where is mate pair information used in sequence assembly process to merge contigs into scaffold even without sequence overlap? Now, some main limitations of Mellon-Pan like metabolic functional inference approaches: severely limited by the availability of appropriate models. A single model cannot work for all environments, for all subtypes of the same environment across different populations, and for all metabolites. Now, Kruskal-Wallis HS is a non-parametric equivalent of ANOVA. That's easy. Now, co-inertia and Procrust are two approaches that can be studied and can study concordance or association between two different multivariate omics corresponding to the same sets of environments. The dash centrality is computed as the number of node-to-node shortest paths that pass through a given node. The answer is betweenness. Now, two examples of why we need to do strain profiling in metagenomic datasets. We need to perform strain profiling to understand specific variations in species that help them to adapt to certain environments, tracking and identifying disease-causing functionalities of outbreak pathogenic strains, identifying novel properties of new strains of the same species, predict engraftment and response in FMT. So these are all the reasons. Two approaches that can be used for strain profiling in 16S data are oligotyping and ASV, amplicon sequence variants. Now, abundances allow an assessment of how well a given species is represented in a community, but does not give the insight into dynamics occurring in the gut microbial community. And a final question is second last question is effects of disease or host-associated other factors can be more efficiently captured by investigating the growth rate in microbial communities. Okay, not the second last one, indeed. Mention two benefits using Spearman over Pearson measure in the line of correlation analysis. So first is non-parametric. Spearman is non-parametric and Pearson is parametric. So Spearman is independent of its distribution, and multiple different types of variables can be compared in Spearman but not in Pearson. Mention any two properties routinely utilized by contact binning applications. So GC%, KMF frequency or TNF, read coverage or abundance, right? In reboot method, that is renormalization, uh, and we also have another, and renormalization, permutation, bootstrapping method. Both In reboot method, both compositional null distribution and bootstrap distribution are compared using pooled samples at test and identify those pairwise relationships that are significant. Number of nodes connecting to the given node is degree centrality. Two usages of network centrality measures, helpful in identifying the central members of the microbiome, can help us to identify hubs or guilds of bacteria, identifying central members, identify hubs. These hubs and guilds can be used as targets in microbiome associated therapies, and functional investigation of these members can help to identify functional dynamics. Walktrap is built on random walks concept, and we have that as the final answer. Now I'll look at some questions that are related again. Which of the following test is used for two group unpaired data and the distribution is unknown and sample size is low? That is Mann-Whitney U test. When we have two group unpaired data, when the distribution is known and the sample size is large, and distribution basically would be since the sample size is large according to the central limit theorem, the distribution would be normal. So in that case, our answer would be Wilcoxon sign rank test. What is the difference between MANOVA and PERMANOVA? So MANOVA is actually non-parametric and MANOVA is parametric. In case of network interference, pairwise relationships after adjusting for certain confounders can be inferred by regression. Now, which of the following approaches is not applicable for associating multivariate microbiome data with multivariate host data? That is PERMANOVA. Confounder is a factor associated with the outcome and the exposure, but not on the causal pathway. Moving on. Which of the following is correct among these options? So statement 1 is reference genome based taxa to gene family mapping method is not efficient for known organisms. That is false. It is efficient. The method mentioned in the above statement is very fast. Yes, this is very fast. That is true. That's the answer. B. Pictus T uses which of the following databases for out reference and genome reference respectively? Pictus T uses green genes for out reference and IMG for genome reference. One species is benefited without harming the others. This phenomenon is commensalism. One species benefits and the other is basically not harmed. Now, which of the following doesn't denote modules in a co-expression network? That is, genes that are randomly distributed across different functional groups doesn't denote modules. Genes that potentially have a direct interaction with each other, yes, that denotes modules. Or genes that belong to the same pathway or pathway modules that cater to production of a specific metabolite, that also denotes modules. And genes that are under the same regulation mechanism, that is also module in a co-expression network. But genes that are randomly distributed across different functional groups, that is not. That's the answer. Now, suppose I have paired samples. Okay, we have made it clear that we have paired samples from 40 individuals undergoing a treatment. One sample is before treatment and one sample is after the treatment, right? So that is paired. I want to check if the abundance of a particular bacteria is significantly altered during treatment. What is the equivalent of Mann-Whitney like non-parametric test that I can use? So that is Wilcoxon sign rank test. Now, the dash between two hyperspaces is the sum of squares of the covariances between all variable pairs. That is co-inertia. That is an important question. Difference between degree centrality and betweenness centrality. Degree centrality is the number of nodes connecting to a given node, right? So number of nodes connected to a given node would tell us the degree, right? For example, we have this as the graph. So degree centrality, let me show you. Let us say this is the graph and these are the nodes, right? So the degree centrality of this one would be 1, 2, 3. 3 is the number of nodes that are connected to this node. This is also 3, then this is 2, then this is 2, this is 1, this is 1, this is 1, and this is 4. This is basically the degree of each node I mentioned near it. And betweenness centrality is for every pair of nodes, there exists at least one shortest path between the nodes. Progrestis relies on the concept of shape correspondence. Two benefits of Spearman over Pearson: non-parametric, distribution independent, and multiple different types of variables can be compared. Now mention two methods used for growth rate prediction, that is peak to trough ratio, grid, and IREP. Now mention two applications of network centrality theorems helpful in identifying the central members of the microbiome, and hubs and guilds of bacteria can be used as targets in microbiome associated therapies. Now, during cell division, the circular chromosome basically forms a replication fork starting from the origin. Reboot method utilized by CREP is devised to address the issue of compositionality associated with the relative abundance data typically observed in microbiome abundance profiles. Topological overlap measure is used to group nodes into different modules like in WGCNA analysis. Two-dimensionality reduction technique methods to visualize variations between strains of given microbe across multiple microbiomes, that is PCA, principal coordinate analysis, principal component analysis. This is PCA and this is PCOA. Principal coordinate analysis is PCOA. Then UMAP, LDA, etc.